Samulya will let us know once we are live. We are just waiting for his thumbs up because he,
Okay. So currently, all the 8 members seven members are there. Yeah. So we are live.
yes. So, yes, I think so. We are live. Yeah, we are already live. Great. Okay. So, thank you so much, sir, for accepting our invite and joining today this event. Uh, so, without wasting much time, we should get into. We are already starting with delay. So, let's get on the event, and I would request my teammate Somilia Mishra to please introduce Professor Samrat Mukhopadhyay today for this event. Somilia.
Thank you.
Yes. So really, thank you, Dhairya, for giving me this very good chance. So it is my pleasure to introduce our today's speaker, Dr. Samrat Mukhopadhyay, a distinguished professor of biology and chemistry at the Indian Institute of Science, Education and Research, Mohali, a highly respected biophysicist, chemist, and Dr. Mukhopadhyay's groundbreaking research dives deep into the molecular mechanism of intrinsically disordered proteins, particularly how these dynamic proteins undergo liquid-liquid phase separation, and sometimes this transition to harmful amyloids process that are central to the neurodegenerative disease like Alzheimer's, Parkinson, and prion disorders. Dr. Mukhopadhyay earned his BSc in chemistry from Jadavpur University, followed by an integrated MS PhD at the Indian Institute of Science, Bangalore, and he did his postdoctoral training at the Scripps Research Institute at the Laola Advanced, and his expertise in single molecule and nanoscale biophysics. Today, Dr. Mukhopadhyay brings his expertise to the psychom synapse. Please join me in warmly welcoming him. Really, thank you, sir, for uh, accepting our invite for the talk. Thank you so much, sir.
Thank you, Samira, for uh, for for having me on your program, which is absolutely an incredible thing that all of you guys are doing, and I'm sure this will benefit the early career researchers across the globe and who want to set up their lab, progress in their career, independent career in science. Thank you.
Thank you so much, sir, for accepting the invite and joining today's our event. That is, we will talk about molecular folding, protein folding, and how it impacts disease biology, and also your journey over the time in the field, and your experience, your rich experience in academia, and uh, your expertise will quite enlighten all of us. Great. So the first question that today I would like to ask you is what inspired you to pursue a career in science? Like, and did you know that you wanted to work in academia when you started with it?
So, yeah, so this is a great question. So, to answer this, I think I have to go back about 3 decades, right?
Mhm.
So, so as you probably know, I grew up in the suburb of Calcutta, now called Kolkata.
Mhm.
And I was also quite, when I was growing up, I was quite influenced by Satyajit Ray. You've heard the name Satyajit Ray, who was very well known for his as a filmmaker, right? And what many people don't know, he was also a writer, and he created some remarkable characters. One of which is very famous detective, fictional detective, and the other one was a fictional scientist, Professor Shonku.
Mhm.
So I grew up reading Satyajit Ray's Professor Shonku, who was a scientist, innovator, and who would travel across the globe and show his innovations everywhere. And I believe there are English translations available of Professor Shonku.
Mm-hmm.
So I think that was one of the early inspiration I got in my almost formative years, I would say, to become a scientist, right? So that was very exciting that you travel around, meet new people, and talk about your discoveries and discuss science with others. So that I kind of got the feeling when I was a kid, right, while reading Professor Shanku. And of course, later on, I had some remarkable teachers, particularly a teacher in chemistry, Bimal Bose, during my schooling. And in Jadavpur University, I had some extraordinary professors. And then, of course, I moved to the Indian Institute of Science, Bangalore, for my master's and PhD program, a very coveted program called the Integrated PhD program.
Mm-hmm.
And there I had the opportunity to interact with some of the really best scientists of the country and across the disciplines, right? And so that really, sort of helped me, in my career. And I knew what I was going to do. And then, of course, I think Indian Institute of Science, Bangalore had a tremendous influence on me. And of course, then I went for postdoc at the Scripps Research Institute in La Jolla in California, actually suburb of San Diego. And I also collaborated with a titan of heat shock proteins, prions, Susan Lindquist, uh, Whitehead. She was at Whitehead Institute at MIT in Boston, in Cambridge, near Boston. And she also was a great inspiration for me. And yeah, so many people influenced and inspired me to become a scientist.
I see. I see. So many people were part of this journey and
Absolutely.
also helped you to shape
Absolutely.
where you.
So as you grow older, you actually realize that your success is not just about you, right? So in the early part of your career, your parents worked very hard for you, right? Your professors, inspire you, teach you, and then your co-workers. And as you become a little bit senior, like when you have a lab, then you realize that you have the opportunity to interact with some of the best students, right? And
Absolutely.
all of them basically shape your career, right? Uh, so today I also draw inspiration from my very talented students, right? Because now I have become more senior. So at this point, I draw inspiration from younger people, like my
Mm.
students. And so therefore, in any journey, including academic journey, you are always grateful that people support you, people help you, inspire you, and you have to just acknowledge that how grateful you are to them.
Absolutely. Absolutely, sir. So maybe since you mentioned now about the students, so maybe you can briefly tell us about, maybe you can share a challenge that you faced during your student years, and also how did you overcome those challenges?
There is an interesting question. I think challenges are always there, right? Regardless of whether you
Mm.
are a student, you are a faculty member,
Hmm.
challenges are always there, right? And you have to always overcome the challenges, or sometimes you have to endure the challenges, and sometimes you just do nothing and hope that the storm will pass, right? And things will be better. So there are many challenges, of course. I think, I mean, there are always specific challenges in research, all of those very specific. But since you asked me, I think to me, I think one of the biggest challenges in my career was my parents and relatives and people around me. See, what happens is when you are a good student in your school, then all of them try to sort of tell you that you should study medicine or engineering, typically at an IIT, right? And very early on, I actually decided to do chemistry. I was interested in taking up an academic career. I was, of course, not sure whether I would become a professor and scientist, but I wanted to do something related to teaching, research. But everybody around me wanted me to become uh, a doctor or an engineer. So I think that was a formidable challenge when I was in my senior schooling and early college, because none of them really wanted me to study science, uh, except my mother, who supported my endeavor into science. Uh, she's the only one I could think of who basically wanted others to sort of not worry about me and wanted me to do the way I wanted at that point. I think that was a big challenge, to sort of go completely against a large number of people, very large number of people. And I can, even at that point, I was kind of absolutely certain they're all my well wishers. They all wanted me to do well, but they wanted me to do something they thought was important. But I wanted to do, chemistry or biochemistry in the later in my career. So I think that was a very big challenge, and that's probably the reason I also wanted to disappear from Calcutta very early on. And that helped me a lot, because when I was a bachelor student at Jadavpur, I knew that I wouldn't be able to sort of make my career in Calcutta, because everybody wants, they say, they used to say, okay, you should have become an engineer, you should have become a doctor, you should have. So that was a big challenge, I think. But that was, fortunately, that problem was fixed very easily by making it to this unique program called the integrated PhD program. In those days, it was very unique, a master's and PhD program at IISc. And therefore, that helped me sort of stay away from people who wanted me to study medicine or engineering. And I could
Hmm.
pursue peacefully scientific, I could actually fulfill my scientific aspiration, scientific dream. And IISc, of course, as many of you know, is an extraordinary place to do science. So I think that was probably one of the biggest challenges that I can think of. But other than that, of course, there are all kinds of other challenges always there. But I think everyone around me later realized that it is important to let the kids do what they want to do, instead of telling them what to do. And it's in the hindsight they know that, but at that time they didn't, right? So
Right.
that was a big challenge. But I'm glad that I could sort of resist that. I could do what I wanted, and and at this point, I can tell you that there is nothing like being a professor, right? This is probably the best profession. If I had to do everything all over again, I would do the same.
Absolutely. I think you are a very absolute example of someone who loves academia and also making academia a better place to
Thank you.
for students. I would definitely
Thank
say
you.
this.
Thank you. I try to,
So,
always inspire students wherever I go, not just here, wherever I go. And they also want to talk to me, discuss science with me, discuss, also ask me about the challenges. So I try to help them. I don't know how much I'm really able to help, but at least they find that there is someone to talk to.
I see. Absolutely. So, since you mentioned about your research interest and you always wanted to pursue chemistry and then later on biochemistry, when did your love started with the protein
Yeah,
folding and
very,
the disease
yeah.
got into?
This is very interesting again. So, when I was in my bachelor's, towards the end of my bachelor's in chemistry, we had some lectures on bioinorganic chemistry. And at that point, I started really liking, studying vitamins, studying amino acids, proteins. Then I started realizing chemistry is what drives biology primarily. And and I started liking this building blocks of life, which comes from chemistry. And I thought, the interface of chemistry and biology would be probably the most fascinating thing to work on. And when I went to IISc, so I started reading more about protein structures. And at that time, in the late 90s, one of the most important questions I found was, how does an unfolded polypeptide chain, which is synthesized as a linear polymer, how does it fold into a very elegant and very unique structure that is biologically active? And I also drew inspiration from some of the PhD theses uh, that I saw in Bangalore, primarily from Professor Balram's lab. And I learned about something called the Ramachandran map. And so GN Ramachandran did some really fundamental work to sort of try and find out the sort of like allowed and forbidden space of a polypeptide chain, right? So which is well known as Phi-Psi Ramachandran map. Then I started becoming fascinated about polypeptide conformation. But I was in chemistry and I was studying something else. I was actually involved in this synthesis of uh, bile acid derivatives, which I was very excited about. As you know, bile salts, they form mixed micelles and they allow fat solubilization. In fact, I synthesized one of the bile acids that is present in birds called avicolic acid. So that was one of the parts of my PhD thesis. And then at that time, it was very clear that if you can combine chemistry and biology, you can learn a great deal about life. And I was, of course, inspired a lot by my own PhD advisor, Professor Uday Maitra, who is an eminent organic chemist at IISc Bangalore. And I was also influenced by Professor P. Balaram. And because if you have paid attention to Balram's career trajectory, he was actually a chemist during his student days, PhD, and then did synthesis, NMR spectroscopy, and then gradually moved into biology. So P. Balram became my obvious role model when I was in Bangalore. And then, of course, so this department at IISc called the Molecular Biophysics Unit, which was actually founded by G.N. Ramachandran in 1970, I believe, when he moved from Madras University to Bangalore. By that time, he already elucidated the structure, triple helix structure of collagen, and he also, introduced this iconic Ramachandran five-side map. And then he moved to Bangalore. Uh, so, so that is the department he founded, and I started interacting with many of my friends from the Molecular Biophysics Unit (MBU), and I was getting very excited about biology. I was learning chemistry, and then I saw there are some beautiful problems in biology. Therefore, slowly, gradually, I sort of uh, became more and more interested in biological problems. And of course, since I did my PhD in organic chemistry, and I did not want to do my postdoc in organic chemistry, I thought at that point I should probably start doing some biochemistry. So before going for a postdoc to Scripps, I actually had about 11 months stint at TIFR Mumbai, Tata Institute of Fundamental Research in Mumbai, as a visiting fellow, which was also kind of unusual at that time. That after your PhD from IISc, which went quite well at that point, and I did not go for a postdoc immediately. I wanted to study biophysics with Professor G. Krishnamurthy, who had a long-standing collaboration with Professor Jayant Udgaonkar at the National Center for Biological Sciences, NCBS, Bangalore. So that sort of gave me a sort of a gateway to biology. So I did a short stint with J. Krishnamurthy and Jayant Udgaonkar, and that was the time I started working on proteins and studying protein folding, aggregation using time resolved spectroscopy. And then I went within a year when I thought I was kind of ready to sort of embark upon uh, studies in biology. So I went for postdoc in single molecule biophysics. So that's how I gradually made transition. And now, of course, we are much deeper into biology. We are working molecular cell biology, a little bit of cell biology have started recently. And we are also diversifying into C. elegans biology, looking at worm model to look at stress granules and other things. So that's how sort of I made a gradual transition from chemistry to biology. And of course, I must say that in Jadavpur University, I had very good professors, not just in chemistry but also in physics and mathematics. Therefore, that solid foundation also helped me later to sort of address problems using biophysics, more physics approach, because in single molecule biophysics, as you can imagine, one has to have some feeling of physics, right? You need to use physics a lot. So I think that's how I was able to sort of combine physics, chemistry, and biology.
I think I'm definitely sure that it is very difficult to combine all these three fields of science, but I think we have termed this different. Okay, this is physics, this
Right.
is chemistry, this is biology, but
Absolutely.
I think nature has not divided it and kept
Absolutely.
it
That's
different.
it. You said it.
So, since you mentioned about your career and how it progressed and how you transitioned into, protein biology and especially biochemistry and folding, how do you stay motivated in academia, especially despite of so many uncertainties, and you never know how it goes ahead.
Yeah. So the uncertainties are on the rise, right?
Mm-hmm, mm-hmm,
So, but I have to tell you that, any exciting career, there are, of course, some safe career tracks, right? But there are some exciting, challenging and exciting. They come together, right? So, any career trajectory that you think is quite exciting will have challenges and uncertainties. See, uncertainties are not just in academia. If you are associated, just imagine if you are associated with filmmaking, okay, if you are associated with sports, if you are associated with music, just imagine how uncertain the life is, right? So, I mean, all of you follow cricket. You see people who are playing just 2 years ago, they're not in the squad, right? So, people who made enormous success just a year ago, they're not able to even make it to 11, right? Uh, in music and film, and I mean, it has always been there, right? Any exciting, challenging career will have uncertainties. Let's accept it, right? It is not easy. It is very hard. So academia is not the only, sort of uh, career which is uncertain, right? And things are very hard, obviously, because of comparativeness, right? Because there are a lot of good people who you are competing with, right? And so, but if you are at it for a long enough, things can get a little easier, right? If you are motivated, if you are dedicated, if you try things for long enough, if you're at it for long enough, things might get easier, uh, but not always, right? Because the challenges are also on the rise, and uncertainties are on the rise. Funds, there are always, not enough funds, right? Always you come across this situation. There are not enough faculty positions, there are not enough other positions, there are too many people competing for the same positions, right? So I think this particular aspect is probably true in any high profile, any exciting career choice that you take. It is not, uh, you know, only in academia. This is what I have to say.
mm-hmm.
But I think uh, one has to uh, be motivated and try things harder and try things little differently sometimes. uh, Maybe about 10 years ago I wouldn't know whether my transition, I took a big risk, right? Making a transition from kind of sort of traditional chemistry into biology. That was a very big risk I took. I could have failed, right? So sometimes some decisions you take that becomes very critical, right, in your career, and you make that decision because you are interested in doing something and you feel strongly about something that you should do this, And when you think you should do this particular thing and not other things, or not there are other choices, let's say. So when you think something you should do, then obviously you are very motivated to do that. And during uncertain times, I think we all had COVID pandemic, right? That was probably one of the really topsy turvy times for us, right? For all of us, regardless of whether you are a student, you are a faculty, you are everybody, right? Everybody was kind of stressed about it. But I think we got over that, right? Sometimes in a very, when you are going through a very hard time, sometimes doing nothing helps, right? If you do nothing, don't respond to the challenges, the storm might pass and you might be actually comfortable after that. Then you take more, some drastic or some, important decisions. But maybe when you're going through a very tough time, it's probably best not to do anything at that point. Just let the thing pass, right? Let the bad time pass. And then you start doing things when you think you are ready to do that. So, I mean, it works. I think depending on the situation, it works differently for different people, depending on the situations. But if one is motivated and one is determined to do take up challenges, I think it is success is not guaranteed, but there is a reasonable chance that you will be succeeded.
Absolutely. So I think you already mentioned quite well, and you motivated people, all the students, I would say, quite well, that why students should consider academia as a career, and and
Why?
like,
So sorry, Adarya. I think why should one choose academia? I think that question I probably have not addressed yet. Uh,
okay,
so
okay,
maybe I should say it briefly. I think
definitely,
academia
definitely.
is probably the only or probably one of the very few professions
Mm-hmm.
where you get paid for doing what you like to do.
Mm,
You can fulfill your dream and you get paid.
absolutely.
And more importantly, what I've realized as a faculty member, being assistant, associate, and now full professor, but I've realized that academia, of course, you are very excited about, your topic that you are interested in, your research
Mhm.
topic, which is the most important thing for you because that is what is going to sort of drive you in your career. But what I've
Mhm.
realized that one of the most important and most exciting things I would say is in academia is to be able to interact with younger students. I think no profession allows you to do that.
I see.
Maybe there are some similarities with music. If you are in music, of course, you are also driven by your passion. There are some similarities with music, and you probably also train younger musicians, or uh, but I think academia is that way is very exciting that you can interact with generations of students, train them, motivate them, and then you can sort of feel proud about your students when they do well, and not only your own students who are working in your lab, but also you have a chance to contribute. You have a chance to make a difference for students who are either at your institute or at different institute or even different countries. So I think that's something which I think is very exciting. This is very unique about academia, that you have a chance to make a difference uh, and you can take chances on people, and some of them will actually flourish, they will thrive, and then you can feel that you have made some contribution. I think that's very unique about academia.
Absolutely. I think this is the most important thing that you already mentioned, that you are getting paid and you're doing something that you want to do it for
That's
yourself and also for society.
yes.
So so moving ahead to the next question, uh, how do teaching and mentoring students enrich your research journey over the time?
Yeah, I think this is an important question. So, in fact, I have to say that a lot of my research has, in the later part of my career, as an independent faculty member, I would say, has is a result of discussions with my students, teaching, okay, teaching the undergrads, which is an amazing experience because you are interacting with you know students who have just finished their schooling, and they want to learn science. And when you teach them, you have to teach at a different sort of pitch at a different level, right? It is not the same as teaching a PhD course, for instance, right? So when you try to teach them undergrads, particularly, then you also learn, right? You also learn by teaching. Richard Feynman, I think, once said that if you want to learn something, teach it, right? So that's very important. When you teach, you are thinking aloud, right? And sometimes by speaking, you get ideas. You probably understand the problem much better than just reading, or sometimes a lot of these concepts get clarified when you are teaching. So in any detective novel, if you are a big fan of reading detective novels, which I am, because I think as a scientist I'm a detective who is trying to solve a problem, right? So in detective novels, you see a detective always has a friend who the person talks to. You have Dr. Watson with Sherlock Holmes, right? So when Sherlock Holmes actually discusses with Dr. Watson, I think things get very clear, right? Then he gets ideas. So discussion also makes things, your ideas get much more, I would say, it gets much more clarity of thoughts. You get much more clarity in your thoughts when you discuss. So teaching is also a form of discussion because you are teaching, then students are asking questions. You are trying to answer the questions. Sometimes the students are, uh, they ask really brilliant questions, right? Then you try to answer, or you try to think about it later, and that enriches your scientific activities. So I feel that teaching has a very important role in overall scientific pursuit.
I see. So I think I should not say it out loud on this platform, but the statement of detective I also included in my statement of purpose and the motivational letter why I want to do a PhD. And this is what I always kept in my mind, that I'm a detective, but a scientific detective.
Yes.
I think this something like this, it really helps. Thank you so much for the super motivational words, not just for me, but but also for a lot of students who are listening to you today.
Thanks.
So, so since you mentioned about, you know, how do teaching and mentoring students enrich the research journey? So the next question that I would like to ask you here is that what advice you give to the young researchers or students who are maybe in their early career, or some scientists who are just near to starting their labs?
Yeah, I think one of the important things is, of course, the most important thing is you have to have a faculty job or scientist job. And then the next most important thing at that point, a lot of people think it's the startup grant and how much startup money you get to set up your lab, which is very important, by the way, because that's like a seed money which allows you to start your research without having an external funding. But I think the most important thing is to choose the initial set of students in your lab. And as I already mentioned, that you know you will basically, your job is to create a scientific curiosity, scientific sort of so you have to get the students who are interested in science, not just they can do science, right? So doing experiments is very important, but I think it is important to uh, recruit students who are equally excited about doing science like you. And if you recruit the right students who are motivated, who are also very good people who you can interact with, and they will also try to get the best fellow co-workers, right?
Hmm.
So that's very important. And I think it's very important to have a very non-hierarchic sort of setup in the lab, because the best output, scientific output, comes when people are happy. People are people feel they're important for your scientific mission and their scientific mission, and they're all respected. And they're in a supportive environment. Everybody likes that, right? I would like that. So as opposed to being overly anxious about you know about publishing, about getting things done quickly, I think it is more important to set up an nice tight knit lab environment. Okay, very supportive environment, non hierarchic, respectful, yet you can disagree, right? And also students should learn to disagree with their advisors. It should not be sort of it should not be sort of like shouting, yelling, screaming, but students should be able to disagree fearlessly with
Mhm.
their advisors if they think something. Many of my students tell me what's the better way of doing it. Sometimes they disagree with the way I told them to do the experiments, and that introduces another dimension to your scientific pursuit, because you know your students are also smarter. Yeah, they are probably, sometimes they are smarter than the advisor. Okay, I have to be very honest. Okay, and they can bring a different dimension, right? Therefore,
mm,
I think it's very important to take them along, and that helps in their professional growth also. And they should know that it is okay to sort of have differences. It's okay to have disagreement. It is, and we can sort of reconcile the differences. If I think what my student is proposing, if I think it's not right, I will try to sort of reconcile the differences, right? Instead of just dismissing them. So I think that is very important. And if you set these conditions right in the beginning, a good working condition, collegiality, camaraderie between the students and you, and if you become like one of them in the lab, then I think the rest of the things will fall into places. Okay. And it's very important to be sort of supportive, collegial, and friendly to your students. And then, of course, the next challenge is to get funding, get the comparative grants, and those things will all fall into places if you have started a good lab with people who are like-minded, who are motivated in science, and so on.
absolutely. Absolutely. I completely agree on this. And also, it, I really love this point of maybe sometimes disagreeing with the PI is also important, because, not everything goes how it looks from outside. Sometimes, things change with course of time, or with the findings while going in the journey of
And if you
researching.
don't have enough funding for some reason, which is totally possible, right?
Hmm.
If you don't have enough funding, don't ask the questions that will require a huge amount of funding
Of
and then
time.
you complain. You start
Mm,
thinking about a problem, sorry, which can be done with the setup, with the constraints you have. And there are a lot of
mm,
these exciting problems. In fact, one should go back and ask the question how G.N. Ramachandran did some remarkable science in India in the 50s and 60s, right?
yeah.
So, and many of, not just, I mean, of course, G.N. Ramachandran was probably one of the greatest examples post independence, right?
Mm, mm,
Uh, sorry. But there are many examples of great scientists in our country who worked
absolutely.
under tremendous constraints, lack of funding. Maybe when they have performed well, they were able to probably get more research funding. But it was quite
Mm,
possible that when they started, they probably didn't have much funding, but they still did very well. So sometimes the choice of problems is an important, because what typically happens is students, postdocs particularly, you are in a well-funded lab, okay, either in India or abroad when you are working, because that lab has been there for many years, and the lab is asking some really big questions, very broad questions, using a multidisciplinary tools, an array of multidisciplinary tools, and they have resources, and you are used to that when you are a postdoc or a PhD student. But when you start your lab, you have nothing of that sort, right? You don't have large number of tools, or you know pieces of equipment, you probably just have one piece of equipment, and you probably have just very little startup funds to start your research. Therefore, you have to recalibrate your expectations, your problems. Then you may not be able to ask right in the beginning a very big question that will require a huge amount of money and resources. Therefore, I think one has to sort of have the understanding of where you are at that point, right? But if you are at it, as I keep saying, things will definitely get easier if you do things right. If you are doing well, things will definitely get easier, and you will be able to address your dream questions, right, at some point. But maybe not in the beginning, right? In the beginning, you may have to sort of recalibrate. You have to sort of do things which are doable under that condition.
Absolutely. I think in today's days, as you quite mentioned well, that asking also right questions is important according to the setup that you have. If you only have like I believe that if you only have a PCR machine in your lab, you can still do great discoveries,
Absolutely. And when
which are
I started,
physically.
yeah, when I started, I had a fluorescence spectrometer. So first several papers we published just using that piece of equipment, which still
Absolutely.
works actually, that particular fluorescence spectrometer.
Wow.
So 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 sometimes you have to think about that, and right in the beginning you may not be able to, you know, embark on the problems that you thought of, right? And it is possible that within a few years things will get better. And if you get a big funding right in the beginning, then sure, go ahead and start with your big question, but that's not possible for everybody, right?
Mhm.
And definitely I did not get some of the coveted funding, right, that many others got. I didn't get.
Mm.
So I had very limited funding at that point. I mean, institute, of course, was in its infancy. Institute, of course, had some, sort of seed money to start with, but I was not able to get a very decent funding in the beginning, right? But things became a little bit better as time progressed, right? So you don't know. If you get it, then of course you can start with your big question and all that. But otherwise, you may have to, and you also have the responsibility of your students.
Mm.
Uh, so therefore, asking some interesting questions which are doable, which are solvable at that point of time, will also make your students happy. They will get their PhDs and move on, because you might get a big funding after 5 years. That may not help your initial students, right? So therefore, you have
Mm.
to see that your students actually. This is very important, because now you have become basically you are sort of like a pilot, right? So people are dependent on you. Therefore, it is very important that your students should be happy, they should be productive, they should get their PhD, they should move on in their career. They shouldn't wait until you get a big grant before you embark on your dream project. They should be doing something that is of interest, that has some significance, and doesn't necessarily need the amount of funding that you anticipated. So I think that is a very important lesson, an important thing, that depending on what you have, how do you sort of recalibrate your expectations, recalibrate your what you want to do. I think that's very important in the beginning, uh, in the early part of your faculty career.
I see. Absolutely. So since you mentioned about uh like the laboratory setup and everything, maybe now you can also discuss a little bit about that. How can students make science engaging and accessible to others.
Yeah. So I think if you are intrinsically motivated, right, and that motivation will percolate in the form of inspiration to the students, people who are around you, right? They will feel it that you are excited about science, right? And I think it is important that the students get excited about your science, and the students then can also talk to other students and younger students who will then get excited about the science you are doing, right? Because you can also, get people engaged in talking about different kinds of things which you are not doing, but they are very exciting. That's also a possibility, which I do at this point, right? That you can also talk about, other things that you are not directly involved in, right? But there are bigger implications, like, because, you know, I was trained as a chemist. I never studied, uh, traditional biology during my early days. But now I find evolution is so exciting, right? So in biology, if you discuss anything, you have to invoke evolution, right?
Absolutely.
So then of course I have to go back and read about evolution, and anything you are interested in you can read, and then your students get excited, and the students actually go and talk about the science, and also the bigger picture of your science to other younger students like undergrads, for example. And we
Mm.
do get quite a few undergrads attracted in our science, and they do internships, they do sometimes master's thesis, right? Which you also did at some point, and you said in MS, right, in Baroda.
I did my thesis. Yeah,
Yeah. So you get so it is very important for you to get excited your own students, and then your students excite in get involved in excite making others excited about the science you are
mm,
doing. Therefore, you can percolate this excitement of science to a much broader scale, right?
mm, mm.
And sometimes we do some outreach or some training. Then, of course, one of the things we did a couple of years ago. So, for several days, my students and I were involved in teaching students and teachers from various places at ISER Mohali about super resolution microscopy, single molecule microscopy, and so on, and fluorescence, general, biological fluorescence, uh, confocal, super resolution, single molecule microscopy, spectroscopy, and so on. So that was a very rewarding experience, and then my students were also involved, as much as I was involved. So that's how you percolate, right? And then you talk to students, colleagues, and that's how they, the students get engaged in discussion with young. Because the students, your students should typically be able to, inspire students who are younger than them. That's how you create sort of like an inspiration wave, right? That sort of encompasses a very large cross section of students.
Absolutely. Absolutely. I think this was very important. So, during my batch of my masters, I think we were very few people who got opportunity to do our master's thesis outside our institute, and I was lucky, I was one of them. So since you were mentioning about my thesis, I did it in IISc Bangalore in biochemistry department. So I also really loved the campus, and as you completely mentioned very well about the hierarchy and how students motivate the next student. So maybe PIs, they motivate PhD students and bachelor's or master's student in their lab, and then they get out and then they motivate others.
Right.
So
So you have to
I
create
think.
sort of like an inspirational wave, right?
Hmm,
which
hmm,
which which encompasses a very broad cross section, right? I mean, it should not
absolutely.
just be limited to your own lab students. And
Hmm,
now,
hmm,
since I mean, now means for many years, I've been also traveling a lot, right? That gives
hmm,
me an
hmm,
opportunity to inspire a very large cross section of students, right? At various places from Trivandrum. So in ISA Trivandrum, students actually invited me, right?
hmm,
So at IIT Kanpur, students invited me, right? So so when I get invitation from students, and I never decline, I immediately accept the invitation. I might decline the invitation from some well-known person if I'm busy in something else, but I try to make sure if students have invited, I go there. And I've actually never declined an invitation from a student, right? So that also gives me an opportunity to interact with students who are not just at ISER Mohali, but also at different places, different universities, uh, different IITs, different universities, state universities, anywhere, right?
hmm,
I
hmm.
was in Bose Institute, a couple of months ago, uh, and the kind of, I was never at Bose Institute in Calcutta. Uh, so I saw some students were really motivated. They were so excited about the science everywhere. Not just Bose Institute. I visited that gives an opportunity to sort of percolate the inspiration to a larger cross section. And then I go abroad, and there are, of course, students there also get motivated. And
Hmm,
sometimes I meet quite a few large, sometimes quite a number of Indian students and postdocs,
hmm.
and I talk to them. I have a special discussion with them, and they ask me many of the questions which you are asking in different forms, right? So that's how you try to make a difference.
Absolutely. Since, sir, so you mentioned about traveling internationally and also in the India to different institutes. How have this international experience shaped your approach to collaboration and mentorship?
Yeah. So, I mean, I've not collaborated extensively. I mean, most of the science that we have done primarily by our lab, but we have some collaboration in France and also at IIT Bombay, where I'm also a visiting professor.
Mhm.
So, and also we have some but I think what the biggest reward of traveling is you try to learn a different system basically, and of course they also learn from me about the system that I'm familiar with. And at this stage of my career, I think the most rewarding experience is to interact with the students, even I would say in different countries. And especially after my lecture, students come talk to me. We have special discussion session with the students, which I had in different places I've been. So that gives me, I think, much more excitement to talk to the students. And often Indian students or postdocs, they ask me about many of the things you are asking. And when you travel, you try to sort of learn other things, right? Not only the science they're doing, also about the general cultural aspects, right? History of that place, right? Sometimes, if you are in Europe, so you know some of the universities are very old, and there's a very rich history in the university, in the city, uh, in many of the historical landmarks, particularly in Europe. And also, last year I was a visiting professor in Japan. So I had the opportunity to learn. So I was in Japan for 2 months, and I had the opportunity to learn a lot of things, very unique things, science and history and the culture, very unique culture. And you try to make friends, right, in science. And then you discuss science, discuss the way they do science, what are the things that you can do better by learning from them, what are the things they can do better. uh, right, there are many things we have in India are much better than elsewhere. so
Absolutely.
so which I will probably write at some point. I'll put it together at some point in form of some article. I'm also influenced by Balram, who wrote some uh, iconic editorial pieces in current science. Maybe I should do that at some point. Uh, some writing. So there are actually quite a few things you realize in India. They're very good, right?
Mm,
It is not always that the other side is greener. I mean, there are things which they learn from us. Uh,
absolutely.
so we have to. So I think the travel interaction that gives a much broader perspective, and you can always make things better, and and you can make a difference to even larger number of students, right, across the globe. Like, I go to Brazil, there's some really talented students in Sao Paulo and Rio. So amazing science people are doing in Brazil. Unless you go there, you wouldn't know, right? So in Japan, I visited, you know, most of the universities that you are familiar with. So that increases your sort of, uh, I mean, I would say that enhances your perspective, right? Broadens your horizon, and that also benefits directly my students, other students who are at my institute. So it is actually a quite an important activity of a scientist, that to go around and interact with people.
Mm, I see. Absolutely, absolutely. So I think we already have a couple of questions from YouTube as well.
Yeah,
I
and
think
also I have to
participants.
say, if I want to say this, so I've also been I've also been very inspired and influenced by my wife, Millie Bhattacharya, who is
Mhm.
also a professor, assistant professor at the institute in Punjab. And we started interacting during our times at IISc Bangalore. She was also an integrated PhD student.
Mhm.
So I think we have grown together as scientists, right? By discussing. So I did not need, if anybody else at that point, because I could discuss with another scientist, right? So that was also a big, I would say, inspiration and big contribution that, of course, as I said, you need to discuss, right? You need to discuss science, or you need to discuss the way you want to do science. So I think that has also helped me a lot in my career, and
mm,
I'm because I've also
mm.
been tremendously benefited by discussing with her. Uh, because you need to talk to people. Science cannot be done in isolation.
Absolutely. I think we would pitch this as one of the most motivated line.
Thanks.
Okay.
So we can take up a
So
few questions. Yeah.
maybe before taking few questions, I would like to ask if you have a presentation to present.
I don't. I have a presentation, but of course, it'll take much longer time. And
Mm-hmm, mm-hmm,
of course, some of my presentations are there on YouTube, and
mm-hmm,
maybe at
mm-hmm.
some point, we can make one presentation, and you can post it
That would be
separately. And but
definitely
I
so.
can tell you a little bit about what we are very excited about, and
Would
then we
be
can
very
move on to the
nice.
question.
That
So
would be very nice.
yeah, so in the independent phase of my scientific career, which so I became assist I joined the faculty of Isar Mohali as an assistant professor 16 and a half years ago. Okay. So during this a decade and a half, my students and I have been tremendously interested in a class of proteins called intrinsically disordered proteins, or IDPs. And so these proteins, as the name suggests, they're intrinsically disordered, right? And they challenge the tenets of structure function paradigm, right? Because in textbook, in biochemistry textbook, you are taught that one sequence will fold into a unique structure that is responsible for a unique function,
Sure.
right? And this particular paradigm is challenged by this class of proteins. And if you look at human proteome, about a third of human proteome is actually IDPs or IDRs, okay, intrinsically disordered regions, okay. Proteins, they might have folded domains, but they are interspersed with a significant length of intrinsic disorder, okay. So one third is a huge, right? One third of your proteome is huge. Therefore, there has to be very important functions assigned to that. So it turns out that the proteins that we are taught in the biochemistry textbook, they are primarily catalytic proteins. Okay, proteins, they're involved in some catalysis or the other. Okay, and for example, lysozyme, for example, chymotrypsin, right? So they have a unique structure. There is an active side. They do enzyme catalysis. But if you now focus the proteins which are involved in cell cycle regulation, signaling cascade, you will actually see many of these proteins are intrinsically disordered. They're very floppy. They exist as a sort of like a dynamic ensemble of structures, right? And it turns out these proteins, or an individual IDP, is capable of carrying out a multitude of functions, because they can form, because of the conformational malleability, they can form different structures at different point, depending on ligand binding, depending on post-translational modification, membrane binding, and therefore they can do a diverse array of functions. And that helps, especially if you think about a compact genome, and you have a wide range of functions you need to carry out, yet you want a compact genome, then of course you can assign a large number of functions to one protein, right? So that is sort of like the evolutionary perspective. Why, as we have evolved, intrinsic disorder has become more and more, right? You can clearly see the increase in the intrinsic disorder as we have evolved. And the other way of achieving function is, of course, to form what you call as biomolecular condensates or membraneless organelles, right? So these membraneless organelles are essentially, they are very different from conventional organelles like mitochondria, Golgi, and so on. These membraneless organelles are sort of like non-canonical organelles. They can form and dissolve depending on the requirement of the cell, right? So what we would call them as on-demand organelles, like for example, stress granules. When there is stress, the stress granules are formed in the cytoplasm, right? So we are very interested in stress granules, how proteins and nucleic acids, especially RNA in this case, come together to form this kind of membraneless bodies and perform the function. Okay. And one of the very prominent membraneless bodies is, of course, nucleolus or nucleoli in the nucleus, right? They are also sort of like phase separated condensates, protein RNA condensates, right? Then you've got transcription condensates, they can regulate transcription, right? So we have been fascinated about this class of soft, squishy materials, okay? Gel-like materials, which are functional and can also become pathological if you alter the interactions. Then, of course, they actually harden to form aggregates, right? Therefore, biology has to operate at an optimal material property, okay, which is like a soft gel-like material property or viscoelastic matter, right? And any kind of changes in the emergent property of the condensate can lead to the disease, right? So therefore it is imperative to understand what are the molecular drivers of the condensate formation and the maintenance of this membraneless organelles and dissolution of these organelles, right? Sort of like the life cycle of this membraneless organelles that are formed by phase separation of IDPs, IDRs, RNAs, sometimes DNAs like, heterochromatin, for example, and they cannot be there longer than necessary, right? And how to sort of tune the material property to for function, and can we understand what are the things if you alter you get pathological aggregates. For example, many of these aggregates are responsible or at least serves as hallmarks of ALS, amyotrophic lateral sclerosis, frontotemporal dementia, and as you said, even Alzheimer's, probably, right? So there is a fine balance between the physiology and pathology that is being dictated by the molecular properties, molecular language, right? So this is what this is the we try to decipher the molecular language using a diverse array of tools that can range from ultra fast spectroscopy, physical chemistry, all the way up to cell biology, right? I think this is what we have been very excited about. And if you are interested, you can read some of our recent papers. And maybe we can also do a separate presentation on the work that we have been doing, at least in the past couple of years, which we are very excited about. We are starting to work on transcription condensates, which are very essential for because if they are unregulated, you can have cancer, right? Yeah, because you can have aberrant condensates that can turn on the transcription, causing tumor, right? So one can also think about, sort of inhibiting such condensate formation, right? So you can think about, designing small molecules. Therefore, there are some translational aspects of our work. Of course, we are interested in fundamental science, which we are excited about, blue sky research. But if something comes out of it, if we are able to address some practical problems, would be even more excited. And we are now
Absolutely.
trying to sort of look at some of these condensates in the live neurons of live organisms. And C. elegans becomes the choice because it's a transparent organism. You can do imaging. So that is something which we are embarking upon. And we are setting things up so that we can look at some of these granules in a live organism like C. elegans. So we are
Absolutely.
very excited about our current research enterprise, our research journey, the questions that we are asking with my talented students, uh, very motivated students. So we are very excited about the science we are doing. So at some point, I'll be able to talk more about the work that we have been carrying out in our lab.
Definitely, definitely, Samrat. I think we will have a separate event,
Sure.
maybe also featuring your PhD students. Maybe they can show their work a little
Sure,
bit
that would
as
be
well.
great.
Yeah, we can host a side chat. Maybe I will forward this to my team, and they will definitely contact you regarding
Sounds great.
this. And also, and also it seems that the students are quite motivated who are attending. We have a lot of questions continuously popping up on YouTube, but maybe I would not able to take all of them, but maybe few of them which we think would be quite beneficial for us and also for others. So maybe one question that I see from Pooja Sri over here is that, in your opinion, what is the biggest unanswered question in the protein folding or misfolding research that you are doing currently?
So, yeah, our lab was at some point, we were very interested in protein misfolding, folding, misfolding, aggregation. Uh, but now we are more interested in phase separation, and then, of course, phase separation mediated aggregation phase transitions. But yeah, since the question is asked, I think there are quite a few questions, very fascinating questions, which are still unanswered. One of the questions, very simple question, which has been around for more than 5 decades, I would say, maybe, yeah, 5, 6 decades, I would say, that how does a protein fold, right? Of course, now we have AI, we can predict protein structures reasonably accurately. But the fundamental question, how a linear polymer of some letter, right, amino acid connected, and that has the information of folding into a unique structure. I think this is, we still don't understand the mechanism, right? We can predict the structure using AI, using AlphaFold, using many other emerging tools, but we still don't understand how an unfolded, or let's say nascent polypeptide chain knows that it has to go to that state.
Understand.
This is still
Mm,
unanswered. Okay. And
mm,
it is actually a problem which is kind of interface of mathematics, physics, computer science, and biology, and chemistry, of course, because chemistry is at the core of the problem.
mm,
Because if you change the side chain chemistry, the folding will change, right? Therefore, it is a very hard problem, and I don't think we have an answer to this problem.
mm,
The other question, of course, which is associated with this particular problem is, how does protein even misfold, right? And do we know the energy landscape? That can we actually predict that if you make this particular mutation, that
Mm.
like prion, for example, one of the great one of the most fascinating examples, which we are also working on, by the way. So so if you have some disease mutation, the prion actually goes to a beta rich state, aggregated state. Now, can we actually make a prediction? Can we actually make a prediction that if you make this particular mutation, and it's not a huge change, right? I mean, one of the changes I can tell you, which I'm very fascinated about, proline being mutated to L, right? Leucine, and that particular change is dramatic, and it is not even in the folded part of the protein, it is actually in the intrinsically disordered part of the protein, right? So, or there are other mutations. Can you actually make a prediction that this will go to this particular structure? So we don't know, right? And if you paid attention to what is called as prion strains, right? Species barrier and prion strains, you will see that prion protein can adopt different types of conformation, and that gives rise to different kinds of phenotypes, right? So that's also something which we don't understand, how different strains forms different kinds of structures, different kinds of aggregates that gives rise to different kind of disease incubation time, lesion profiles, the phenotype, because the same prion protein might be involved in CJD or fetal familial insomnia, but the disease expression, disease presentation, lesion profile, everything is different, right? So that's something which I find very exciting to think about.
Absolutely. I think this is something really unanswered, and I'm working in the field of cell death, and I was just curious in between the event, and I was looking at which are the intrinsically disordered proteins in cell death, and it seems that maybe very important proteins in the field of cell death are
Yeah,
intrinsically
absolutely. And any
disordered,
cell
and
cycle regulatory proteins, you will see there are
yes,
intrinsic disorder, and they are there for purpose, right? They are doing
yes.
some really critical function, which is not achievable by the traditional folded proteins, right?
Absolutely, absolutely. So maybe we can quickly take one more question. And I think one question that is really interesting. So Kriti has asked this question, and she asks that can misfolded proteins act as a molecular memories of past cellular stress and
Yes,
influence the future stress response?
yes. Uh, there is, of course, there is a line of evidence in
Mm-hmm.
this particular aspect. In fact, I told you that I worked with Susan Lindquist,
Mm,
who was basically an iconic figure in the field of heat shock proteins, prions. Uh,
mm,
so she and Eric Kandel, so they published a paper during the time I was doing my postdoc. So it must be around 2005 and 2006. So they published a paper showing the effect by taking a model protein from Aplysia snail.
mhm,
This is a protein called CPEB, cytoplasmic polyadenylation element binding protein, which has a prion-like domain. Actually, very significant length of this protein from Aplysia has prion-like domain, which forms amyloids, but beneficial amyloids. Okay, so they are involved in local mRNA translation, local
mm,
translation, and molecular memory. So
mm,
Eric Kandel and Susan Lindquist's paper had shown that, and there were subsequent series of papers in different kinds of models, including Drosophila, uh, the CPEB, which is called, which has a different name in Drosophila, ORB. So they're involved, apparently they're involved in male courtship memory. So the formation of amyloids. Now there is an emerging consensus that there might also be there might also be a contribution of phase separation of these
mhm,
proteins. So yes, amyloids, as you can imagine, amyloids are beautiful nanostructures and very ordered nanostructures. So we know amyloids, they're involved in Alzheimer's, Parkinson's, alpha-synuclein amyloid, tau fibrils, filaments, and but amyloids, since it's a very robust, well-defined structure, obviously Mother Nature has used amyloids for some beneficial purposes, right? One of the purpose, of course, there are, we know, I mean, I worked on yeast prion protein, which also forms beneficial amyloid prion in yeast, Saccharomyces. uh, Basically, under stress condition, it has a different phenotype. It's sort of like an epigenetic phenotype. So it you have the same gene sequence, but under different conditions they have different phenotype that is coming from prion formation, right?
mm, mm,
So non-Mendelian you know inheritance. So that is also responsible, that similar mechanism is also responsible for memory formation, at least
mm.
uh, part of that long-term memory formation. Okay, so yeah, amyloids and many of these things might have been used for beneficial purposes.
Okay.
Thanks for asking that very interesting question. Yeah.
I think I was also not aware about this. If misfolded proteins can also have memory, this is
Yeah, it
something
can
which
it
is.
yeah, it can play a role in memory, long-term memory. Because
Mm,
often this is a fascinating question, right? I mean, the life of a molecule is not that long, but we remember things which are, like decades ago, 2 decades, 3 decades ago, right? Or 5 decades ago. People who are in their 80s, they remember things that happened when they were in their, teenage, right?
mm,
So this is a fascinating question actually to think about, that
mm.
molecules don't last for that long, right? Because there is
Oh,
always
dynamic.
uh, formation, and there is always a cycle. Molecules are destroyed beyond a certain point, right? But how
Mm,
you're retaining the memories, that's something which I think is one of the most fascinating questions in neuroscience, that how
mm,
synaptic plasticity, one of, in
mm,
fact we are interested in C elegans here. You can study the plasticity very in a tractable fashion, but again C elegans is probably not the best model system to study memory that is retained for decades, right? Then of course one has
mm,
to go to much higher organisms.
mm.
So it's actually quite a fascinating question. The molecules are gone, but still you have the memory. So amyloid might be one of the mechanisms.
Mm, yeah, definitely possible. Good. Then I think we will have to take an end to this event. I think it's prolonged, and we, I mean, it's really nice that we have huge response from students and they are asking a lot of questions, but we
I
are
must
extremely
thank.
sorry.
I'm not able to see any of them, so I must thank all the students and postdocs who joined. uh, and I think some of the things I said might be of some help to you. If not, at least it was a pleasure interacting. Not interacting, I couldn't interact with all of you, but thanks for joining. And I'm also reasonably active on Twitter. Particularly, I also post the academic activities, scientific activities, and travels associated with science on Twitter. So you can always follow me on Twitter. And I'm sure I'll probably see some of you in the future, maybe in the very near future. And looking forward
Okay.
to seeing some of you.
Definitely, definitely. So, so we have a unique concept also of a side chat. Maybe we are also taking that ahead a little bit now. So there we invite especially PhD students and postdocs to come and discuss, the topic of interest and in the science, and also where they are working, and also we are like there. All the participants are allowed in the Google meeting, and they can directly interact. So due to some security concerns here, we have kept it on YouTube. But maybe we would invite some PhD students from your lab, and then
Sure,
we can discuss the
sure.
science overall with them.
Yeah,
Absolutely. So
my students
first
would
of all,
love to
thank you
do
so
that.
much.
Yeah, my students would love
Absolutely.
to do that. Yeah.
Definitely, sir. So first of all, thank you so much for accepting our invite quite readily.
And you should
Uh,
call me, you should call me Samrat next time whenever we meet. So that's how
Definitely,
I'm called by my all my students.
definitely, Samrat. So I would love to come to Isar Mohali as well
Absolutely.
when I'm back
It's
in
a great
India.
place. You must come.
Absolutely. And also, I would like to thank the audience today who are here, staying with us, and taking their time, and that to Sunday morning time, lazy days, and coming and listening us.
Thank you all.
I will thank you so much, sir.
Thank you
Great.
all.
Then we take an end to this event. There are a lot of unanswered, unasked questions, which we would definitely consider in part two of
You can,
Samrat
can you actually
Mukhopadhyay.
keep these questions and then we can discuss at some point and
Definitely,
you can
definitely,
answer to them? Yes. Okay.
definitely. We
Good.
can, we can, we can keep these questions,
And
and we would also
the
direct.
people who ask the question, their names also, so that we know who asked the
Definitely,
question. Great.
definitely,
Awesome.
sir. We can
That's
forward
great.
them to you as
Okay. Kanvi says done.
well.
Kanvi, you will also
Thank you
call
so
me Samrat when we meet. Okay. Yeah. Okay.
Okay. All right.
Okay.
Goodbye. Have a
Take
good
care.
time, sir,
Bye bye.
Samrat,
Bye
and
bye. Take care.
goodbye,
Bye. Thank
everyone.
you. Bye.
So we have our feedback form in the chat box. Please do like, share, and subscribe, and please do fill up the feedback form to get for your getting your certificate as well. Thank you.